one-dimensional explanation and then a diagram, and then we'll quickly, because biology these days is all three-dimensional. So you would be able to see how the virus is going to get into the cell and which will multiply, right? So what virus does is first it attaches. So these molecules are present on the surface of the cell, and this is one of the viral proteins. Remember this umbrella-like structure that I showed you before, correct? And these are complementary to each other. If you look from three-dimensional structures, these are complements. So they fit to each other like lock and key. So it's called virus attachment. Once the virus attaches, then it penetrates into the cell. Once it is inside the cell, the protein shell is removed, because all of us have proteolytic enzymes. It's the same darn thing that people use it as meat tenderizer. You see, what it does is it really chop up the proteins into the smaller pieces. So this protein is chopped up. Once the protein has been chopped up, now the nucleic acid molecules are naked. And once the nucleic acid molecules are naked, this RNA is released, right? There was the enzyme which will convert this RNA into DNA. And see, this is again pretty neat thing, because for the long period of time we thought the genetic information only reside in the DNA. Not true. See, there's no DNA here. It's an RNA. Polio virus is RNA. Rabies virus is RNA. So genetic information can reside in the RNA too. And then this RNA is chewed up again. You left with a single stranded DNA, which is converted into double stranded DNA. And then it integrates into our own DNA, our own chromosomes. Okay? Now here's another thing. If it integrates, it has two possibilities. Whether the virus is going to replicate, right, or not from this cell where the virus has been integrated. See, if you look at the incubation period of HIV infection, and incubation periods means the time when the virus gets into your body till you get the first set of symptoms. It's called the incubation period. Usually it is few weeks, okay? But the longest incubation period that I have seen in the literature is 15 years. Okay? So there is no way on earth, no medical laboratory can certify that a given person is free from HIV infection. Absolutely no way. I'll give you one example. See, say I somehow get HIV, and this HIV got into the memory cells. Remember immunological memory concept? And say this memory cell was directed for malarial parasite. That this cell will going to respond only to malarial parasite and to nothing else. And this cell will only divide when I'm exposed to malarial parasite. Otherwise this cell will not divide. It's a memory cell, it is going to stay there for a long period of time, correct? So I got it in down Kalamazoo, right? When I retired after millions of years, and say somehow I thought, okay, now I want to go and see some safari in Africa, and a female Anopheles mosquito bites me and puts some malarial parasite into my body. Now that cell will start dividing. The moment it divides, the virus will come out. If it doesn't divide, it will never come out. So it's really, there is absolutely no way to figure it out whether some person is free from HIV or not. And this is the same, explanation how the virus is going to get in and how it is going to get out. But I will show you the three-dimensional various computer.
It's down below. It's hit escape.
See? Huh? No, let's go. Yeah, okay. You are absolutely right.
Am I doing this?
So this is only 5 minutes, yeah? I can create within.
Oh, okay, very good.
So this is just 5 minutes. Everything that I said, you can see in 3 dimensions.
The replication of HIV-1 is a multi-stage process. Each step is crucial to successful replication and is therefore a potential target of antiretroviral drugs. Step 1 is the infection of a suitable host cell, such as a CD4 positive T lymphocyte. Entry of HIV into the cell requires the presence of certain receptors on the cell surface. CD4 receptors and co-receptors, such as CCR5 or CXCR4. These receptors interact with protein complexes, which are embedded in the viral envelope. These complexes are composed of 2 glycoproteins, an extracellular GP120 and a transmembrane GP41. When HIV approaches a target cell, GP120 binds to the CD4 receptors. This process is termed attachment. It promotes further binding to a co-receptor. Co-receptor binding results in a conformational change in GP120. This allows GP41 to unfold and insert its hydrophobic terminus into the cell membrane. GP41 then folds back on itself. This draws the virus towards the cell and facilitates the fusion of their membranes. The viral nucleocapsid enters the host cell and breaks open, releasing 2 viral RNA strands and 3 essential replication enzymes: integrase, protease, and reverse transcriptase. Reverse transcriptase begins the reverse transcription of viral RNA. It has 2 catalytic domains: the ribonuclease H active site and the polymerase active site. Here, single-stranded viral RNA is transcribed into an RNA-DNA double helix. Ribonuclease H breaks down the RNA. The polymerase then completes the remaining DNA strand to form a DNA double helix. Now, integrase goes into action. It cleaves a dinucleotide from each 3' end of the DNA, creating 2 sticky ends. Integrase then transfers the DNA into the cell nucleus and facilitates its integration into the host cell genome. The host cell genome now contains the genetic information of HIV. Activation of the cell induces transcription of proviral DNA into messenger RNA. The viral messenger RNA migrates into the cytoplasm, where building blocks for a new virus are synthesized. Some of them have to be processed by the viral protease. Protease cleaves longer proteins into smaller core proteins. This step is crucial to create an infectious virus. 2 viral RNA strands and the replication enzymes then come together and 4 proteins assemble around them, forming the capsid. This immature viral particle leaves the cell, acquiring a new envelope of host and viral proteins. The virus matures and becomes ready to infect other cells. HIV replicates billions of times per day, destroying the host's immune cells and eventually causing disease progression. Drugs which interfere...
So see, it's far easier to see than listen to somebody, right? So this is, and I think remaining few minutes, I will talk about the epidemiology and the disease itself. This is a typical picture. Somebody, so what we are doing in this figure here is we are looking at how HIV is replicating in a patient, right? These are the days after infection. These are different names for the different stages of the disease which have been given. And here I'm measuring how much virus is present in the blood. Okay? So I'm measuring the protein which is making the shell of the virus particle. It will directly tell us how much it is. And in the beginning, if you see, for the first few weeks, the virus is replicating very well. Then for a few months, it totally disappears and it reappears. So it was really puzzled for the long period of time where the heck virus is going. Okay? But we knew